Hello! On behalf of my co-authors and myself, thank you for attending my talk on sexual dimorphism in the Bony Labyrinth. There are numerous successful methods for using human remains or skeletal material, in this case, for estimating the sex of an individual. If we have both a complete skull and a complete pelvis, we can estimate the sex for that individual with up to 80 to 90% accuracy. However, we encounter at least 4 problems when putting this into practice. The first challenge I'll talk about is often the case with archaeological remains. The skeleton will be incomplete or damaged. Another challenge is that with subadult individuals, their skeletons do not exhibit the sexual dimorphism of adults because they have not gone through puberty yet. A third challenge we face is that features are often population specific. For example, we can have populations where both men and women tend to be shorter, or we can have populations where both males and females tend to be taller. If we find an individual and are not familiar with which of these populations that individual comes from, if we had a taller individual, we could falsely estimate the sex as male when in fact this individual could just be a taller female coming from the overall taller population. A fourth challenge is when working with hominins, they tend to be poorly preserved and overall more robust than modern humans. So this research is filling a gap where we're looking for a well-preserved bony element that has to be sexually dimorphic and not population specific, as well as age independent. Let's talk about the Petrus portion. The Petrus portion is the hardest bone in the mammalian body. It is considered the most often recovered bone at both archaeological sites and at crime scenes. You see here I have the temporal bone highlighted in green, and on the inside we see here with the green outline the Petrus portion. The Petrus portion is so robust it is even able to survive cremations. So why am I talking about the Petrus portion? Well, housed safely within this robust, well-surviving Petrus portion is the Bony Labyrinth. We can see here this is a Petrus portion that has been made to be more transparent so we can see the Bony Labyrinth safe inside. What is the Bony Labyrinth? The Bony Labyrinth is the internal ear and consists of the semicircular ducts, the anterior, lateral, and posterior, as well as the cochlea. During life, the Bony Labyrinth is filled with the membranous labyrinth. But afterwards, and in archaeological contexts, we're working with just the Bony Labyrinth, which is a cavity, a hollow, empty cavity. First, let's talk about the cochlea. The function of the cochlea is for hearing. We can see here in this cochlea spiral that the spiral gets tighter as we move towards the center. Towards the center, this cochlea is processing low-frequency sounds, and where the spacing is wider, we see that this cochlea is processing high-frequency sounds. Cochlea morphology varies between species based off of what that species needs to hear in order to survive and to communicate with other members of that species. On the right we have a humpback whale cochlea, specialized to hear a wide variety of frequencies. And on the bottom we have a chinchilla cochlea, also specialized but to hear a very specific frequency. Now let's talk about the semicircular canals of the bony labyrinth. The inner ear has 3 semicircular canals, an anterior, a posterior, and a lateral canal. These are responsible for maintaining balance during movement and head movements. The semicircular canals differ between species, with larger canals being associated with higher sensitivity as well as agility, such as faster movements, head movements, and canals with a smaller radius are considered to be less sensitive. On the left, we see the bony labyrinth of a bush baby with very large, sensitive semicircular canals, useful for not falling out of the trees, followed by a land-dwelling whale ancestor, an ancient whale, and a modern dolphin. As these whale ancestors are moving from the land into the sea, we see that their semicircular canals are getting smaller and less sensitive. And in the dolphin, we see this unique extension on its cochlea. This is used to process the dolphin's echolocation. The bony labyrinth is fully adult shape and size before birth, actually in utero, by 17 to 19 weeks in utero. Our research is building upon research by Osipov et al. That found sex determination was successful using the bony labyrinth with up to 84% accuracy. They used discriminant functions, where you put in your measurement multiplied with a set value plus a constant to get your value. The research aims for my doctoral work then was to see if sexual dimorphism existed outside of what Osipov et al. Found in 2013 with a Greek sample. Does it exist in diverse samples? I would expect that it would. And interestingly, I would also expect there to be the same sexual dimorphism in subadults, since the bony labyrinth has been found to be adult shape and size in utero. And could it be possibly useful for human evolution applications? Morphology should be diagnostic. For this research, I expanded upon Osipov et al.'s work on sexual dimorphism in a Greek sample and included a sample from Germany, from Illinois, and South Africa. These geographically diverse skeletal samples needed to have adult, known sex individuals from recent modern humans. I had 16 males and 14 females for a total of 30 Norris Farms Aniata individuals from Illinois. The date is AD 1300. Then a total of 38 Zulu individuals from South Africa, and these date to the early 20th century. And then a total of 30 German individuals from collections in Baden-Württemberg, and these are from the 19th century. So overall, I had 50 males, 48 females, for a total of 98 individuals for this study of sexual dimorphism in the bony labyrinth. My methods included taking the known sex individual crania or temporal bones and scanning them in micro-CT scanners on site at the locations where the skeletal collections were housed, then using Aviso medical imaging software to create 3D models of the micro-CT scans. Next, I virtually extracted through segmentation. And we can see here the process of removing the Bony Labyrinth from the surrounding Petrus Portion. Through segmentation, I was removing that cavity and extracting it for study. And this allowed me to have a 3D virtual endocast reconstruction of the Bony Labyrinth in each individual, right and left. And I was using Aviso software to do this as well. Following a method used on the Bony Labyrinth by Guntz et al. From 2012, I added a wireframe skeleton through the 3D reconstructed virtual endocast. The skeleton is calculated by thinning the structure's volume down to a wire. Then, instead of placing 2D linear measurements for height and width, I placed landmarks, here pictured as the yellow dots, on the surface, and used the skeleton as an anatomical guide, and then used the Pythagorean theorem to calculate the distance between certain sets of landmarks that correlated with traditional linear measurements. Since I placed landmarks instead of 2D measurements, I have more information, more data that can be used in future studies. I also have the information about the shape of the canal outlined in green lines. We can see here why I didn't want to just stick with more traditional measurements of height and width for each canal, because although these two objects here both have the same height and the same width, their shape is quite different, and thus the use of landmarks so that I have that data preserved as well. Let's look at the results of a look at cross-population sexual dimorphism in the Bony Labyrinth. With all samples, the German, Zulu, and Anyata lumped together, we applied the function 1 from Osipov et al. 2013, which was a Greek sample, and had an original success rate of 82.4%, to all our samples together, and this had 69.39% success at estimating male or female. We created a new function, new discriminant function, based on the three samples together, and this had 71.4% success at estimating sex. Looking at all the samples together in a t-test revealed that 14 variables had significant sexual dimorphism between males and females of 21 variables. And the posterior semicircular canal was the most dimorphic variable when all samples were lumped together. Overall, with all samples together, we see a trend towards females having smaller bony labyrinths and males having larger bony labyrinths. Looking specifically at the German sample, when we apply function 1, we see that the success rate is 66.67% for estimating sex, and a new function based just on the German sample, this discriminant function, had 70% function success for estimating male or female. And in the German sample, there was the least amount of sexual dimorphic or significantly sexual dimorphic variables found in the t-test, with only 2 being significantly sexual dimorphic for the Germans. And for the German sample, the posterior semicircular canal, again, was the more sexually dimorphic variable. In a separate analysis using only the Zulu sample, application of function 1 had 68.42% success, and a new function created had 71.1% success at estimating male or female in the Zulu sample. And in the Zulu sample, we see much more sexual dimorphism in the t-test than the Germans, with 10 of the 21 variables being significantly sexual sexually dimorphic. Also, amongst the Zulu sample, the anterior semicircular canal tended to be the more significant variable for sexual dimorphism. For the Aniata sample, an application of function 1 had the highest success rate. Again, this is based off of a Greek sample that had 82.4% on that Greek sample, and when applied to the Aniata, the success rate was 73.33%, estimating the sex of the individual. Within the Aniata sample, we created new discriminant function, and this had 80% discriminant success. So this is equal or equivalent to having a complete pelvis and a complete crania present, but it is just from small measurements within the Bony Labyrinth. The Aniata also had the most number of significant variables in the t-test for sexual dimorphism. 13 out of 21 were sexually dimorphic significantly, and with the Aniata, the Cochlea tended to have the most sexually dimorphic variability, with females having different shape, more rounded, and males tending to have a more taller or height-wise tall cochlea. This is a principal components analysis, or a PCA. We see the German females are in pink, the German males are in red, the Zulu males are in dark blue, the Zulu females are in light blue, the Aniata females are in light green, and the Aniata males are pictured in dark green in this PCA. Our PC1 has 50% of variance, and PC2 16% of variance. And as we move towards the right, we see larger size being determined. So the Germans had the larger size of the three samples. As we move along this axis of the PCA, we see that as we move upwards, the shape of the Bony Labyrinth canals tends to be more vertically longer, and as we move downwards along this PC, we see that the shape of the Bony Labyrinth canals and cochlea tends to be more horizontally broad. Let's take a look at a summary of our findings. We were looking for sexual dimorphism in diverse samples, and our outcome shows that we do have sexual dimorphism in geographically diverse samples, similar to what Osipov et al. Found, but they are population specific. And in the notes section, we see that when we look at populations, there is a difference in shape. And when we look at sex, we see differences in size in the Bony Labyrinth. Future work ideas include having more modern human samples, subadults, or increase the fossil sample size, and more shape and volume work, as well as primate samples. At the bottom, you see a few of wireframes I've created from my Bony Labyrinth landmarks. The bottom has a male extreme or a female extreme, or we could compare the samples, the Zulu sample extreme to the German extreme. Lots of different directions to go with future work. Thank you very much for listening to my talk. I look forward to seeing you at the next conference, and thank you to the Leakey Foundation for funding this research.